The CRISPR Cas9 system is a tool for cutting DNA at a specifically targeted location. The technique has revolutionized gene editing. There are two components to the system, a DNA cutting protein called Cas9 and an RNA molecule known as the guide RNA. Bound together, they form a complex that can identify and cut specific sections of DNA. First, the Cas9 has to locate and bind onto a common sequence in the genome called PAM. Once the PAM is bound, the guide RNA unwinds part of the double helix.
Once it finds the correct sequence, Cas9 can cut the DNA. Its two nucleus domains each make a nick, leading to a double-strand break. The cell tries to repair the cut segments of DNA, but it is prone to introducing mutations. This makes CRISPR good for taking out specific parts of the DNA. CRISPR can also be used to make specific edits. To do this, one or two Cas9 cutting domains are deactivated. Cas9 then transports the enzyme to the specific DNA sequence. This causes mutations in specific DNA bases. Some examples include mutating a disease, causing sequence to a healthy one.
CRISPR could also be used to promote gene transcription. Here, Cas9 is completely deactivated and transcriptional activators are added to the complex, fusing directly or with a string of peptides or recruited to the guide RNA. This brings transcription machinery like RNA polymerase and other factor targets to promote transcription. To silence genes, a crab domain is used to fuse Cas9, inactivating transcription by recruiting more factors that physically block the gene. CRISPR can also be used to observe the 3D architecture of the genome by attaching fluorescent protein complexes to map where specific sequences are found in the genome.